So we've all heard the name CRISPR, right? It's been sold to us as one of the single greatest breakthroughs of our time. But what if, in our rush to unlock all that potential, we also unlocked something, well, something far more dangerous?
I mean, the hype was just unbelievable. The promise was nothing short of a revolution, an end to genetic diseases, a brand new era of medicine. It really felt like we had reached a new peak of human achievement.
And this was the big idea, the metaphor that captured everyone's imagination, that we could finally go into our own DNA and edit it, like a text document, correcting the little typos that cause so much pain and suffering. It's a beautiful, powerful, and very clean idea. But this is where we have to pump the brakes for a second, because the latest research is suggesting that the risks aren't just about making a few mistakes here and there, those off-target effects. No, the real danger might be baked right into the very way CRISPR works, a fundamental feature, not a bug, in the system we thought we could control.
Okay, Section 1, Chaos inside the cell. To really understand these risks, we've got to start small. I mean, really small. We're going to zoom all the way in, past the body, past the tissue, and right into the nucleus of a single cell. That's where our DNA lives.
So we were sold this idea of genetic scissors, something clean, precise, almost surgical. But the biological reality, it's a lot messier. This slide really lays out the difference between the promise and what's actually happening. See, CRISPR isn't really a pair of scissors. It's more like a targeted demolition charge. Its whole job is to break our DNA, and then it relies on the cell's panicked and often chaotic internal repair crew to try and put everything back together.
And that chaos? It actually has a name, Chromothripsis, and it is just as violent as it sounds. It literally means chromosome shattering. This isn't a neat little snip. It's a catastrophic explosion where an entire chromosome gets pulverized into tiny fragments.
And this is how that cellular catastrophe unfolds. First, step 1, CRISPR makes its cut. Now, this can cause a piece of that chromosome to get lost, walled off from everything else, which you see in step 2 inside its own little bubble called a micronucleus. Now, inside this isolated bubble, step 3 happens. The chromosome shatters into bits. And finally, in a desperate attempt to fix things, the cell randomly stitches these pieces back together in step 4. The result? You get this monstrous, rearranged chromosome that might have just lost its most important cancer-suppressing genes, or, even worse, amplified the very genes that cause cancer in the first place.
All right, Section 2, the patient's dangerous paradox. So let's zoom out from that one cell to the actual person receiving the therapy, because here, the dangers get even more complex and honestly, pretty counterintuitive.
And this is the absolute crucial question. What if the very act of trying to fix one faulty gene creates the perfect storm for a far, far worse disease to take hold? This slide explains the p53 paradox perfectly. Take a look at the left side. In a normal, healthy cell, you have a protein called p53. It's known as the guardian of the genome. It detects the DNA damage from CRISPR and basically orders the cell to self-destruct, so the therapy fails. But now look at the right. In a precancerous cell, one where p53 is already broken, that cell just ignores the damage and keeps on living, multiplying like crazy. So the therapy is actually acting like a filter. It kills off the healthy cells while selecting for the survival of the most dangerous ones. It's a huge problem.
And there's another problem. The Cas9 protein, the scissors part of CRISPR, it's derived from common bacteria like Streptococcus, the thing that causes strep throat. And since most of us have already been exposed to these bacteria, our immune systems are already primed to attack Cas9 on sight. This means a therapeutic dose could trigger a massive inflammatory response. Or, even worse, our own bodies could hunt down and destroy the very cells we just paid a fortune to have edited, which would make the entire treatment totally useless.
Okay, Section 3, editing the human species. So now the scale of the risk blows up from a single patient to our entire species. And this isn't some what-if scenario, this has already happened. Back in 2018, the world was absolutely stunned by the announcement of the first gene-edited babies. But if you look past the massive ethical firestorm, what you see is a technical disaster. The edit didn't work consistently, which created a mosaic of edited and unedited cells in the children's bodies. And what's even more alarming is that the gene he targeted, CCR5, isn't just a doorway for HIV. It's also involved in our immune response to other viruses, and it even plays a role in brain function. We have permanently changed these children's biology in ways we simply do not understand.
And this, of course, opens up a profoundly disturbing social question. Once this technology is widely available, who's going to get access to it? It threatens to turn the economic inequality we already have into permanent biological inequality. You're creating a world of genetic haves and have-nots.
Section 4. Rewriting the rules of nature. Now we're going to zoom out to the widest possible lens, the entire planet. Because CRISPR isn't just for humans. It can be used to alter whole ecosystems with a technology called gene drives.
The most important thing to understand is this number, 100%. Normally, any given gene has a 50-50 shot of being passed on to the next generation. A gene drive hijacks the rules of heredity, forcing a specific trait to be inherited by nearly 100% of all offspring.
So what is a gene drive? You should think of it as a genetic chain reaction or maybe a genetic wildfire. Once you release an organism with a gene drive into the wild, that new trait is designed to spread like fire through the entire species. It's self-propagating, and once it's out there, it's potentially irreversible.
And if you're sitting there thinking, oh, we're smart enough to predict the consequences, well, history has a very humbling lesson for us. On Macquarie Island, scientists removed invasive cats to save the native birds. Seemed like a great idea, right? But the rabbit population, which the cats had been eating, absolutely exploded. And they ate the entire island bare, causing a total ecological collapse. This is what's called a trophic cascade, and a gene drive could trigger one of these on a global scale.
And this brings us to our final, and maybe our darkest level of risk, the deliberate weaponization of this incredible technology.
With the rise of these massive genetic databases, we now have detailed maps of the subtle genetic differences between different groups of people. If you combine that knowledge with CRISPR, it becomes theoretically possible to design a pathogen that only targets people with specific genetic markers. You could effectively create a weapon that attacks one ethnic group while leaving another completely unharmed. It's no longer science fiction.
And the possibilities here are just terrifying. Researchers have already proven they can rebuild extinct viruses like horsepox, a cousin of smallpox, completely from scratch using mail-order DNA. A government or a terrorist group could do the same with smallpox itself. Or they could use CRISPR to give common bacteria resistance to all known antibiotics, or just make the seasonal flu way more lethal, engineering a pandemic that our modern medicine would be powerless to stop.
But the threat doesn't just come from sophisticated government labs. Maybe the single biggest risk is that this incredible power is becoming available to almost anyone.
This table really just lays out the divide perfectly. On the left, you've got professional science, operating with strict safety protocols, ethical review boards, and years of training. And on the right, you've got the world of DIY biohacking, where often you're just dealing with a garage. The tragic case of Aaron Trawick, a biohacker who died after he publicly injected himself with an untested therapy, is a really stark warning. This combination of boundless ambition and very limited expertise, it's a recipe for disaster.
So we really have to ask ourselves, from the chaos inside ourselves to ecological collapse, from engineered inequality to accessible bioweapons, CRISPR has undeniably opened a box of immense, almost unimaginable power. The question that is going to define our future is, do we have the wisdom and the foresight to control what we've just unleashed?